So we're focusing in on photosynthesis in this section of the unit, and we've already talked about what this word means, but we want to remind ourselves. Photosynthesis means to make. And remember, what we're making are organic compounds. Now, prior to studying photosynthesis in AP Bio, we just said that photosynthesis makes sugar. Except we have to remember that not all plants are actually storing that as glucose or sucrose or sweet sugar. These organic compounds can be used as energy sources, or they can be used as structural components, like cellulose. Now remember, starch is actually an energy storage molecule. So the point of photosynthesis is to make organic compounds. The way that we make an organic compound, remember, to be organic, it has to have CHO, carbon, hydrogen, and oxygen. Now there's plenty of carbon in the air, the atmosphere, carbon dioxide, but that's an inorganic molecule. It's not an organic compound. So the point of photosynthesis is that I use carbon dioxide and water to build these organic compounds. The way that we do this overall is an endergonic reaction. I have to put energy in, and the energy source initially was light. However, up to this point, we've actually taken the light. Remember, during the light-dependent reactions, that was the first stage of photosynthesis. During the light-dependent reactions, we took the light energy, and the light first excited the electrons. Then those excited electrons pumped hydrogen ions, and then chemiosmosis produced photophosphorylation, which gave us ATP. So we've taken the energy from light and stored it into ATP. But we have another energy storage molecule because this electron actually ends up on a carrier, NADP+, which gets reduced once it accepts that electron to be NADPH. So the light-dependent reactions is where we take the energy from light and store it into two temporary sources, NADPH and ATP. And of course we call this the Z scheme, and we can use cyclic photophosphorylation to make more ATP. Because what we're going to see is that we need more ATP than we do NADPH. This ATP and NADPH are going to be used to build glucose. But we need to stop for a second and remind ourselves where in the plant this is happening. It's great for us to know the term thylakoid space and know that that's where our hydrogen ion gradient is high. That's the thylakoid space, the stroma, the thylakoid membrane. Here's our chloroplast. We've got our chloroplast here. And inside the chloroplast are these stacks of thylakoid membranes. Each stack is called a granum, and the space around the granum is called the stroma. So that's great. We should know this from knowing the light-dependent reactions. But we really also want to know overall where in the plant this is happening. So we go back to this picture here, and we remind ourselves that in a plant, the light-dependent reactions can only occur where there is light. So the main photosynthetic place of the plant is actually going to be the leaf. Main photosynthetic is the leaf. Now when we look at leaves, they look pretty flat, pretty nondescript, right? There's a bunch of cells there. But it turns out that these cells are actually organized into structures that we need to know. The top is called the epidermis, and this epidermis is a clear coating. Why clear? Because we want the light to be able to penetrate it. But this coating helps protect the rest of the cells. It also helps trap water. Now remember, we need water at the very beginning to be our source of electrons. The water, of course, is absorbed by the roots, transported up to the leaves via something called the xylem, or the vascular bundle. And we're going to need to know this term a little bit later. This vascular bundle is what carries water up from the roots and delivers it here. Now the main cells that are going to be performing photosynthesis are called the mesophyll cells. This is where there's the highest rate of photosynthesis. These cells are going to have lots, dozens, hundreds of chloroplasts. And these cells, the palisade and the spongy, are where most of the photosynthesis is going to happen. Now the point of the spongy mesophyll is so that there is space for gas storage. Coming into this area will be CO2, so CO2 will enter, and O2 should exit. How is it going to enter and exit? Well, in our bodies, gas enters and exits through, like, our nose and our mouth, holes. Guard cells in the plant are actually going to form a hole called a stomata. And if you did your lab report on cell transport, you know that the stomata helps regulate the entrance and exit of carbon dioxide, but also water loss. When that hole is open, water can evaporate. This can be good because that evaporation of water helps pull other water up behind it, but it can also be bad if the plant loses too much water. So we do need to know a little bit about the structure of the leaf, because where the light-dependent reaction happens is where we want our Calvin cycle to happen as well. So these cells are going to be doing the light-dependent and what we call the light-independent reactions. So we're here, we're in the leaf. The leaf got some light. It was delivered water. And we started the light-dependent reactions. So photosynthesis, remember, starts with the breaking of water using light. And these reactions, the light-dependent, results in an NADPH, an electron carrier, and ATP. These are energy sources inside of the chloroplast, but in the stroma. So we're in the stroma. We're now going to do what's called carbon fixation. Fixation means to take from an inorganic form, inorganic, to an organic form. So we're going to take our inorganic CO2 and we're going to fix it onto another molecule. You do need to know the name of this molecule. It's called ribulose bisphosphate, but you can just measure, remember, RUBP. RUBP is a 1, 2, 3, 4, 5 carbon compound. I add 1 CO2 onto it, and it will become a 6 carbon compound. However, this cycle actually only happens 3 times simultaneously. So I actually need 3 RUBPs, 3 CO2s. I will get 3 6 carbon compounds, but that will then break into 6 3 carbon compounds. Each of these 6 carbon compounds is very, very unstable. So the 6 carbon then splits into 2 3 carbons. 2 times 3 is this number, 6. So a little bit tricky there. I start with a 5 carbon, I add a 3 carbon, that splits into, I start with a 5, I add a 1, 5 plus 1, 6. 6 divided by 2, 3's. But this has to happen 3 at a time. So that's how I end up with 6 3-carbon compounds. To my 3-carbon compound, I'm going to remove each of those phosphates. And that's how I make, to my 3-carbon compound, I need to add phosphates. So I'm going to use 6 ATP. Then I'm going to take my electrons and I'm going to oxidize NADPH. I'm removing the electron. This means that I'm actually reducing my carbon compound. So I've attached carbon, I've attached more phosphates, I've attached more electrons, and now we get this molecule that we've seen before, which is called glyceraldehyde 3-phosphate, or G3P. This G3P can be used by the cell. Now the reason that this has to happen 3 times in a row is because I'm only going to get 1 G3P for every 3 carbons that I put in. Now it would make a lot more sense if the cell could just take 3 carbon dioxides and push them together to make a 3-carbon compound. But it turns out that's not how these enzymes work. These enzymes work by putting 1 carbon dioxide onto a 5-carbon, giving us a 6, splitting it into 2 3's, then adding more phosphates, then adding more electrons, and this happening 3 times simultaneously to give us 1 G3P, because now I have to recycle the rest of these back into 5-carbon compounds. So a little bit tricky in that I'm slowly adding on carbon dioxides, and I'm only getting 1 G3P out of this. If you can see, we're divided into 3 phases. Carbon fixation, add the carbon, reduction, add the electrons, and regeneration of our UVP. You'll notice that we do need to use 3 more ATPs. So this means that we have actually total used 9 ATPs, 6 NADPHs, and we have made 1 3-carbon compound. We did use 3 CO2s as well. So we used 3 CO2s, 9 ATPs, 6 NADPHs, and 3 CO2s, and I've made 1 3-carbon compound. You'll notice we've recycled our electron carriers. You'll notice that we have our ADPs ready to be used again during the light-dependent reactions. And we've made a G3P, 1 3-carbon. Now I can add 2 3-carbons together to make glucose, and I can put bunches of glucose together to make cellulose or starch. But the Calvin cycle, which you need to know, is that it makes 1 G3P for 3 turns. 3 turns of the Calvin cycle makes 1 3-carbon compound. 3 turns uses 9 ATPs, uses 6 NADPHs, uses 3 CO2s, makes 1 3-carbon compound. So it's a little complicated to track the carbons, but the big point is that you're adding 1 CO2 onto a 5-carbon. This is happening 3 times in a row, and out of those 3 times I get 1 that I get to keep. The rest of these get regenerated into our 5-carbon compound. Just a quick little preview. Moving forward, we are going to talk about this enzyme Rubisco that is supposed to fix the carbon and sometimes gets distracted by oxygen. So our next lecture is going to be about Rubisco and something called photorespiration. For now, focus on understanding what the Calvin cycle is.